I'm very pleased to welcome our next speaker, Professor Sharon Peacock, who's Professor of Public Health and Microbiology at the University of Cambridge, a renowned expert in pathogen genomics and antimicrobial resistance over many years, but also Executive Director of the COG-UK consortium that's provided the world-leading sequencing of the SARS-CoV-2 virus. And I think I'm right in saying, Sharon, I haven't checked it in recent weeks, but at one point, on the Gavi website, it was definitely the number of sequences from the UK outweighed the sequences from the rest of the whole world.
Pretty much.
Wonderful.
For that introduction. Thank you. Thank you so much. Well, Sarah's a hard act to follow, and thanks again to her for her incredible contribution to the COVID response. So I'm going to change gear, as you've heard, and I'm going to be talking about how we use viral sequencing to generate actionable information. This isn't about stamp collecting, this is about trying to apply the information we get to public health interventions. Now I'm going to split my talk into three. The first is really looking at how the past prepared us for the present. We were one of the first countries in the world out of the starting blocks to generate sequences in such high volume, and people would say to me frequently, how on earth did you do that? So I'm going to describe the building blocks that allowed us to really get started very early. I'm then going to describe how the UK actually generated the genomes at scale. We've sequenced more than 1 million genomes now, which is completely unprecedented in the history of sequencing. And then I'm going to go into some case studies where we look from achievement to actionable information. And I'm going to put that onto a timeline of the pandemic from the beginning to now. So let's look at how we were ready to start sequencing as quickly as we were. The first is that there was incredibly strong UK engagement over more than a decade to explore the potential for pathogen genomics. In the same way that we heard from Sarah, she'd done a lot of work beforehand to prepare the ground for a new vaccine. We had been working for more than 10 years to look at how we could use sequencing for public health benefit. And that was across the spectrum of academia, public health, etc. Now, we also had support at high levels of government. So back in 2013, you may remember that David Cameron made an announcement around the 100,000 Genomes Project. Now, we heard a lot about the cancer genomes and rare diseases genomes. What you might not have heard about so much, it's quite deeply buried, was a theme around pathogen genomes. I chaired that theme of work, and we recommended that we should be routinely sequencing TB, hepatitis C, and doing deep sequencing for HIV, so we could look for minor variants that could be resistant to antiretroviral therapy. So the government were aware of pathogen sequencing. Then that work was passed on to PHE, as it was then, to upscale, and that is now in force. Genome sequencing of foodborne pathogens is absolutely routine now. That's often how we detect outbreaks. And then going on to Dame Sally Davis, her Volume 2 annual report in 2016 called A Generation Genome, she got us to write a chapter on how sequencing can be used against bacteria and viruses. Around the same time, actually, academia and public health agencies were gaining experience during outbreaks, MERS, Ebola, and Zika, and this was often on the ground. There was also huge investment into the Wellcome Sanger Institute, and the Medical Research Council had funded something called CLIME. That's a cloud infrastructure for microbial bioinformatics. That's where you can put sequence data and get it analysed very, very quickly, as well as the Arctic network, which was thriving, and that's for methodology. So that was the complete building blocks for what we did next, which was to develop the COVID-19 Genomics UK consortium, which emerged from this legacy. Now, if you look at our response, it's been called exemplary, and I believe that that's the case. We didn't sequence the first genome. That was sequenced by Chinese researchers and released, as you heard earlier, on the 11th of January, which informed vaccine design, and the virus was characterized in a paper in Nature in early February. However, we were the first to develop a capacity to generate genomes at scale and use this for public health impact. And the pandemic has really shown beyond any doubt that sequencing generates actionable information that supports an effective response. That includes designing vaccines, keeping vaccines effective, looking at the emergence of resistance against other therapeutics such as monoclonal cocktails, monoclonal antibody cocktails, looking at the emergence of variants of concern, looking at those transmitting around the world, and looking at transmission in closed environments. So how did COG-UK begin? Well, it actually began with early support from the government chief science advisors of Patrick Vallance and our CMO, Chris Whitty, who discussed the importance of genomics to the pandemic response. So on the 4th of, you see a timeline along there from beginning of March to the end of March, and the green blocks are the genomes that we generated, the red blocks are the positive tests at that time, of course, an underrepresentation because there wasn't sufficient testing available at that time. So on the 4th of March, having been encouraged by Patrick Vallance, I actually sent 5 emails to colleagues saying, can you phone me this afternoon? And in those phone calls to expert informaticians, I said, look, I think we're going to have to set up a genome sequencing capability. Do you agree? Is this, am I kind of away with the fairies? And they said, yep, we think that's important. So within 2 days, by the 6th, 20 emails had gone out to experts around the country to say, please meet next week at the Wellcome Trust. Jeremy Farrar was present at that meeting, and on the 11th, 20 of us got into a room and we created a blueprint for a brand new entity called COG-UK, where we decided how we were going to sequence, where we were going to do it, who was going to do it, and how we were going to network that together. That was an extraordinary meeting because scientists will often talk about the latest findings, but we were talking about the nuts and bolts of how to put together a genomics consortium. By the 15th, we had a proposal submitted to Patrick Vallance, and that was funded quite soon through the COVID-19 Fighting Fund. And in the meantime, we got underway with our sequencing. We assembled teams to focus on key areas that included sample collection, method development, linkage to metadata, analysis and report writing. And even before we were funded by the 23rd of March, we'd written our first report to SAGE. Alongside that, we had to further develop Climb COVID, and then COG-UK finally commences on the 1st of April officially. Now, looking at our purpose and vision, actually it was spot on. Our purpose was a research collaboration that generated scientific evidence to demonstrate that SARS-CoV-2 sequencing can bring about benefit to public health. This was unusual. It wasn't for research purposes. It was researchers looking for the benefit that they could generate. So our vision was really to establish an end-to-end sequencing capability and demonstrate added value of providing this information to our public health colleagues for action. So this is what we formed. This is the network. So we had 16 regional sequencing sites, which were in academic institutions and public health agencies. We had a central sequencing hub at the Wellcome Sanger Institute, together with our Climb for our big data. We had project administration in Cambridge, and of course, significant funding, particularly over time. Now, one of the really unique things about this was that we were able to bring together a four-nation approach. So it was a United Kingdom effort, which hadn't been done before with genome sequence data, and that was critical for how we use the sequence data. So we had a consortium agreement which allowed data sharing signed by the 4 public health agencies and academic institutions across the UK. That was no mean feat to get 21 different institutions to sign up to one legal agreement. Now, I'm going to talk about our achievements, looking at the phases of the consortium. You saw this earlier, and what this shows at the top is the number of cases diagnosed from the gov.uk website. As we heard JVT say earlier, the first phase is underrepresented because of our testing capability. On the bottom, you've got the number of genomes we generated, and that's on a log scale from 0 to 1 million. And I'm going to divide it up into different phases, as you'll see as I progress. Now, the launch was really getting from 0 to 10,000 genomes, getting things off the ground. Now, colleagues have said it's like trying to take an airplane down the runway, still assembling the wings on it and hoping that it's going to take off. This was a time of really pushing on to do this rather than worrying about perfection. It was very much output and method development. So our focus really was around, for example, sample collection and operations and logistics. So if you can just imagine, for the first time, we had to link 105 NHS laboratories testing so-called Pillar 1 samples to 16 different regional sequencing hubs in academia, together with networking the Lighthouse Labs, which are testing community samples, Pillar 2, with the Wellcome Sanger Institute. It was a logistic headache. We had to develop methods for SARS-CoV-2 sequencing at a good cost and at pace. We also needed to work out how to link the data and the metadata together. Don't forget that in the early days of testing, if you ever went to a lighthouse lab, pre-lighthouse lab in testing in the community, there may be somebody with a clipboard collecting information about you, and that's very difficult to collect their metadata. It got better over time. And then, of course, we had to focus on analysis tools. There's no good gazing at the genomes for hours and days and months and then telling somebody after that period of time that you found something. You need to do that in an automated way within a matter of minutes or hours. And analysis tools were extremely important. And in fact, if you look now, looking back at the tools and methods we created, they are used worldwide. So our sequencing methods are used extensively together with our Pango lineage nomenclature. So Andrew Rambo and colleagues were critical in designing that, together with a whole host of bioinformatic tools from both Andrew Rambo, from the Birmingham group, from Oxford, and from Cambridge and Glasgow. These tools have allowed us to analyse the genomes and make them work. So moving on to delivery, which is really June to November 2020, delivery 1, we reached 50K at the end of this phase, and that's about 50% of the global total at that point. Now, what do we do? Well, in that period of time, our data and tools were used in more than 120 retrospective and live outbreak investigations in the UK alone. Our analysis was actually very varied, but we did multiple activities. For example, we looked at importations into the UK. And if you look here, this is a beautiful work which shows that in the first wave, we were particularly worried about China. We were particularly worried about northern Italy in terms of importations. But what we found actually was that a third of cases were introduced from Spain, about a third from France, and about a third from Italy. And so this allowed us to start to understand importation patterns, and this information was used by the Border Force. We also looked at epidemic waves in Wales, Scotland and England. We looked at the spread of the so-called summer holiday variant B1177, which spread from Spain. We looked at outbreaks in hospitals and care homes, and we particularly looked at mutations, which we know is so important. It's what everybody is thinking about in the viral genomics field at the moment. So this is just some examples. In Cell, Emma Thompson in Glasgow, she looked at a particular mutation which showed that it was N439K in the spike protein, which showed immune evasion. But one of the very first examples of that sort of type of biology being reported, together with D614G in the spike, it became completely prevalent in the population over time. So this is the D version, this is the G version. It became dominant. And this allowed us to test out our methodology. So we knew with this it was slightly more transmissible. This graph shows actually there's no difference in outcome of patients with either the D or the G version. We were sharpening our pencils, getting our methodology really ready for when we really needed it. So this was some of the work that we did. We did a first UK science showcase, which has had 62,500 views on YouTube so far. Then Alpha came along, and that's when everything changed. It was the first variant of concern, so-called VOC. As you know, detected in the south of England. The earliest genome was actually on the 20th of September 2020, but with a very high growth rate through to December, when the initial analyses indicated that Alpha had rapidly acquired 17 mutations, which is quite unexpected. Now, there's a particular amino acid substitution which defined it, which is N501Y in the spike, which changed the in the receptor binding motif of the spike, which increases binding affinity to the human ACE2 receptor. And this had been found recently by a colleague in South Africa in another variant called Beta, and actually putting heads together, we realized that this was going to be important in Alpha. So what do we do? Well, from the genomes, we were able to demonstrate that this was more transmissible than previous variants that have been circulated. We demonstrated there was no significant impact on vaccine efficacy. We showed an increased risk of hospitalization, but no increase in death once admitted, and we were able to demonstrate this affected diagnostic assays but did not alter their overall outcome of positive or negative. So this is what sequence data is has the power to do, because it categorizes the viruses into different groups, which you can then apply different studies on. You'll see here a paper on the 9th of December from Andrew Rambo, submitted to Virological Org, which described this lineage. This is just a few examples of the work that we produced at that time. So I've talked about assessing transmissibility of SARS-CoV-2. So that's Eric Volk's Imperial. Looking at the estimated transmissibility and also impact on patients in England. We worked very closely with the Oxford Vaccine Group and provided sequencing for their vaccine trials. So we're also included on their study of alpha and looking at immune evasion properties. Now, this is what we call the Grinch variant, because actually the impact on COG-UK was very large. Christmas was cancelled for many of us, unfortunately. I remember sending out an email to everybody about a week before Christmas saying, thank you so much for working so hard. Thanks to your families, we've done a great job. And we had a skeleton staff over Christmas. And about 3 days later, I was writing back to them saying, really sorry, but we're all going to have to come back for Christmas and start sequencing because we need to up our outage. So there was a rapid increase in demand, an expectation for variant surveillance at this point, a rapid increase in media requests. It actually became extremely uncomfortable for a period of time. And during that period, we progressed from 100 to 2,000 genomes by mid-January. At that point, PHE established a very important variant technical group to have a formal process of monitoring these variants. And we developed bilateral contracts between COG sites and PHE to upscale through additional staff and equipment. Delivery 2 was into this year now, February through to June 2021. We increased up to 500,000 genomes, and our major focus then was really reducing our turnaround time, getting our information to end users. We had a second showcase that was viewed by people from 49 different countries. Now, I've said we're focusing on turnaround time, and this slide shows that we could now sequence more genomes quicker and cheaper. So, for example, here you've got median number of days from sample receipt at the sequencing site to submission of sequence metadata to our database. And what you'll see is that we got quite good, and it's between 2 and 3 days. The time it takes from the time that a patient has a swab to the data being generated is actually longer, but it's now around 5 days. There are some parts of that process that we don't have any overall control on, but I think that that's quite remarkable to bring those turnaround times down. You can see here the cost of sequencing. So we more or less halved the cost of sequencing over the last 18 months of SARS-CoV-2 through our efforts. And you can see the number of genomes sequenced and deposited. These are some examples of our delivery 2 phase. It's slightly fuzzy, but you can see here that we're looking at if I can read it even myself, apologies if you can't, but certainly looking at transmission in universities. That's just been accepted today in Nature Communications, looking at how the virus spreads between university students and whether they are drivers for community infection. Does it go out into the community as a result? This is looking at COVID-19 in care homes. We also looked at transmission in hospitals, and this very important paper here described a very first a multi-centre interventional clinical study to look at the impact on infection control of sequencing the virus. That's being led by James Blackstone and Judy Brewer, and they're analysing that data at the moment. And again, Delta, when everything changed. So we know quite a bit about Delta now. It's first detected in India at the end of last year, spread to 98 countries rapidly within months, and now is everywhere. It's got some mutations in the N-terminal domain, receptor binding domain, and furin cleavage site, and it's been assessed to be around 50% more transmissible than alpha. It demonstrates partial immune evasion, which we heard about from Sarah earlier, and particularly in relation to the first dose. And by this point, we were up to 1 million genomes. So you can just see this extraordinary graph here of the variants that are circulating over time. You may be familiar with this, but this is the summer holiday variant, which actually had no particular properties. It wasn't a VOC. It was simply spread by people travelling, actually, their holidays. And then we have Alpha, and then we have Delta. And I haven't talked about Delta Plus today, but I can answer questions on that if you're interested. In this phase, we were still looking at neutralisation of Delta together with changing composition of lineages over time. We were also publishing reviews for other people on variant spike mutations and immune escape, together with ongoing efforts to increase our methodology or improve our methodology. Transition. Now, we're researchers. This isn't our stock in trade. And so from April to September 2021, we had a planned handover to the public health agencies so that they did the routine sequencing. Now, that's more or less done and dusted. We form a safety net so that we can do surge sequencing, and the sampling strategy is now set by Department of Health and Social Care and UK HSA. As I say, we are there if a backup is required, with Wellcome Sanger Institute doing a very large amount of the sequencing now, with their capability up to around 50,000 a week. What about going forward? Well, that's going to be the content of another talk, but what we really need to do now is start to reanalyze our virus genomes in the context of human genomes and very detailed metadata, so we can understand much more about disease causation and outcomes for patients. And there are many other things that we're actually planning to do. So scientific publications, we have generated around 60 papers which have already been published. So this represents actionable information for the world. So I'm just going to move on now really to thank people. So this is a team effort. There are 600 people in our consortium, mostly volunteers. We want to thank our funders and funding facilitators. You can see here who funded us, but each of these people listed here have been critical in us getting the funding. We initially had funding to get us started, but we needed further funding to keep the fires burning. And we've now developed a training program for overseas colleagues so that we can support training and sequencing. It's called CogTrain. Partner organisations have been absolutely brilliant. So there are 16 of these, together with our public health agencies and the Central Sequencing Hub. Every one of these have been absolutely stellar. These are all the people we collaborated with in addition. So we have other academic partnerships together with the NHS. As I say, this was quite an extraordinary team effort, and without them, we wouldn't have done this. We also have extensive science collaborations. So we sequence for all of the national core studies. We underpin that, including for REACT. And we're now collaborating with many other people to try and link our genomes with other types of data. This was a team built from scratch, and that was possible because we had an incredible management team, which is the top 2 rows. We also had an incredible steering group, which is the bottom 2 rows, and they kept us safe, they kept us working, they kept us effective. And so this is like a big business, really, developed on the hoof. And finally, these are our COG UK members. I'd thank both them. And this is important. This list is really important because when our affiliation goes on publications, and we have a publications policy, this is the list of names that go on there to honour their contribution to the work that we've done. I'd also like to thank COG UK member families because they have actually put up with quite a lot whilst we've all been working very hard. So thanks to all those family members. So that's it, and I'll take any questions.